Hey, welcome friends. In this video, I will be discussing about the bioinorganic chemistry, which is the most requested topic. And I recently came home and I don't have the whiteboard with me right now, but I will manage it up at the end of this week. So today I will make the video with pen and paper. So let's start today's topic. So from the bioinorganic chemistry, the major topics which we have to do for our CSI NET exam or GATE exam, that things I have listed below. So first I will talk in a little introduction that what topics we have to do. So the major topics that we have to do, you can see, is that the metalloporphyrin proteins, the metalloporphyrins or the heme proteins. In these headings, the things that comes are the electron transport proteins and the oxygen transport proteins. For the electron transport proteins, the cytochromes are the important things we have to do. So there are several type of cytochromes, and later we will discuss in detail about them. So in electron transport protein, we have to do the cytochromes. For oxygen transport proteins, we have to do the hemoglobin and the myoglobin, their functions and their structural features. Now after that it comes the non-heme proteins, and for the non-heme proteins, they also show the same activity as shown by the heme proteins, the cytochromes and the hemoglobins. So in non-heme proteins there are also electron transport proteins and oxygen transport proteins. For the electron transport proteins, the main important things are the iron sulfur clusters or iron sulfur proteins. They are ferredoxin and rubridoxin. And then the blue copper proteins, for their blue colors they are called blue copper proteins, and we will discuss all about them in later in details. And then the non-heme oxygen transport proteins are the erythrin and hemocyanin. These two things we have to discuss. Then after that we have to do the photosynthesis, about the chlorophyll, the manganese cluster and the overall photosynthetic process, the scheme we have to discuss. Then after that the enzymes, these are very important. The carboxypeptidase, what are the structure of the enzyme, what is the catalytic cycle, so what are the catalytic cycle and what is the active metal in the enzyme, that we have to discuss. So then the carbonic anhydrase, the aldehyde oxidase and vitamin B12, these four major enzymes we have to study. And after that the nitrogen fixation, this is very important. In vitro and in vivo nitrogen fixation, this is a very important topic. So then comes the storage protein. We already discussed about the transfer protein like oxygen transfer and the electron transfer, but we also have to study the storage protein, and they are the ferritins. And the last thing is the cisplatin. So these are the main topics we have to discuss while reading the bioinorganic chemistry. If I miss any other thing, I will obviously discuss later while I will explain these things in detail. So first I will start from the heme protein, the hemoglobin, myoglobin and all other heme proteins. That thing I will discuss first. And for that you can see, this is the structure of porphyrin. So this macrocyclic compound which contains pyrrole rings, you can see these are the pyrrole rings, 4 pyrrole rings are there, and these are this is called porphyrin, and this is a structure of free porphyrin. So this free porphyrin can give away 2 protons to form dianion. So dianion will be there, N minus and N minus. After deprotonation of these 2 hydrogen, there will be 2 negative charge. And then if you give a metal 2 plus cation, that will be incorporated into the porphyrin ring, and it will give you this metalloporphyrin complex. So this metalloporphyrin complex is called the heme unit, and there will be other substituent in these positions, but you don't have to worry about that. You can't remember all the substituents. So this is the basic structure of metalloporphyrin. Now both cytochrome and hemoglobin, myoglobin, they are the metalloporphyrins. As you can see, I wrote down that both the cytochrome and hemoglobin, they are metalloporphyrins, but they have some basic difference that I have to discuss. That hemoglobin and myoglobin, they are oxygen transport proteins, whereas cytochrome is the electron transfer protein. And why this difference is that will be clear from their structural features. So we have to look into the structural features of the cytochrome and hemoglobin and myoglobin. So first I will discuss about the cytochrome C. So sorry, about the cytochrome. Now there are three type of cytochromes mainly, and they are cytochrome A, cytochrome B, and cytochrome C. So these classification is dependent on the nature of the substituents. I already mentioned that there are several substituents here, and I will not discuss about the substituent pattern. You can find it in any book, and this is not important at all because you cannot remember all the substituents and no questions will be given from there. But you should know that according to the substituents, they are classified as cytochrome A, B, and C. Now cytochrome C is the most studied cytochrome, and about it we have the most information. So we will discuss about cytochrome C. So you can see this is the structure of cytochrome C, and as you can see the four coordinates. So iron here will be octahedrally coordinated, and four coordination sites are occupied by these four nitrogen atoms from the porphyrin ring, and the fifth position is occupied by the nitrogen atom of the histidine residue, and this protein chain is also linked with this porphyrin itself. And another coordination site will be coordinated by the sulfur of the methionine, and this is also linked to the protein chain. Okay, so this is the full structure of cytochrome C, you can see. Now the structural features of cytochrome C tells that all the coordination sites of the iron is fulfilled, so there is no vacant position for accommodation of any other ligands, and that's why it cannot accommodate or it cannot accommodate oxygen like the hemoglobin or myoglobin, who are the oxygen transport heme, and they can accommodate oxygen because they have vacant position. But in this case you can see all the six positions are occupied, but they can change their oxidation state. So in this case iron may be +2 or +3 oxidation state, and actually they shuttle between this +3 and +2 oxidation state, and that do the electron transfer mechanism. So the main work of the cytochrome C, the main function of cytochrome C is the electron transfer, because their coordination centers are fulfilled and they cannot do electron transport. Now what is the mechanism of the electron transport in cytochrome is shown by this scheme, that first the electron is transported from cytochrome B to cytochrome C, and then from to cytochrome A, and finally it is transported to oxygen, and this oxygen is reduced now. So by the chain function of this all type of cytochrome, finally the oxygen is reduced. Now it shows that to function like this, to do this mechanism, the cytochrome A should have some ability to bind with oxygen, because the cytochrome A will bind the oxygen and finally transfer the electron to it. Now that means it should have 5 coordination number rather than 6, because if it has also 6 coordination number, then how can it occupy the oxygen? So it will have 5 coordination, and that's why it can accommodate oxygen. And these cytochromes are responsible, and these cytochromes which are called cytochrome A type and which has vacant site, they are actually responsible for the toxicity of cyanide. That we say that cyanide is very toxic. Cyanide means Cn minus. They are very toxic to our body, and the toxicity is due to the cytochrome A, because in cytochrome A iron is in +3 oxidation state, and cyanide, so this cyanide when binds to this iron 3, they are very tightly bound, because cyanide is a very strong field ligand, and when they bind to cytochrome cytochrome A with iron 3 oxidation state, it stabilizes the complex, and then the iron 3 cannot return to iron 2 state. So the shuttling between iron 2 and iron 3, that stops. There is an enzyme called cytochrome C oxidase which can oxidize this so that can oxidize the cytochrome. So iron 2, as we have seen that iron 2 and iron 3 are shuttling between the two in the electron transfer process. So cytochrome C oxidase is that enzyme that oxidizes the iron 2 to iron 3, and then again iron 3 is reduced to iron 2. So this is how the mechanism works, and you have to just know about the structure of the cytochrome C oxidase, but the full structural features of the cytochrome C oxidase is unknown. Only which is known is that it contains cytochrome A3 and cytochrome A, and also two copper atoms which are called copper A and copper B. So this is the structural features of the cytochrome C oxidase. And one thing I have to mention about the cytochrome C, the toxicity of cyanide, is that one misconception is that the cyanide can also bind with the hemoglobin, and it is one of the reason for the toxicity, but that is not true, because you know, in hemoglobin the iron is always in +2 oxidation state, and cyanide ion, which is a negatively charged species, a hard center, that has a very less tendency to bind with soft center like iron 2, and that's why it cannot bind with iron 2 in hemoglobin. So the main toxicity of cyanide is due to the fact that it bind with the cytochrome C and not to the hemoglobin. So I will finish the video here, and in the next video I will discuss about the hemoglobin and myoglobin.